Hi, welcome to the seventh tutorial of the Genomics to Proteins portal. Genomics to Proteins portal, aka G2P portal, is a powerful discovery tool to link genetic screening outputs to protein sequences and structures. G2P has two modules, where either you can look up your gene or protein of interest, or you can analyze your own mutation, annotation, or protein structure data. In the previous tutorial, we have explored the interactive mapping module, start with the gene protein identifier, to introduce how users can navigate through various protein features, structures, and genetic variants interactively with user-provided annotations. In this tutorial, I'll introduce you to another exciting part of the interactive mapping, starting with your own protein structures. Whether you have unpublished in-house protein structures or the latest computational structure predictions beyond AlphaFold, this model allows you to upload any protein structures and interactively map with your annotations, even extending beyond the human proteome. We'll be looking at a case study from a published paper on protein-protein interactions in MLA lipid transport system in gram-negative bacteria. The authors performed unbiased mutational scanning on MLA-C protein, which is one of the core elements within the MLA lipid transport complex, and mapped the mutational hotspots of MLA-C to the AlphaFold predicted protein complex structure. The author provides a spreadsheet in the supporting information for MLA-C mutational hotspots, along with the residue number and amino acids, whose average fitness change upon mutation is meaningfully larger than the mean fitness of all mutations. Along with this fitness score, the authors annotated these mutational hits on the crystal structure of the MLA-C monomer, so let's take a closer look at those hits on the structure. You can download a crystal structure of MLA-C monomer in the presence of phospholipids from Protein Data Bank, and then upload the downloaded file to the interactive mapping module with the corresponding chain identifier. By loading the structure, it will preview the MLA-C crystal structure binding with the phospholipid. Next, you can upload your annotation data. From July 2024 release, G2P now allows users to upload directly from a CSV file, or you can copy and paste your data into the window. So let's enter the mutational scanning data here. After submitting, you can check your annotations if there's any formatting errors in your data. The module will automatically detect your data type for you, but users can still change it whether it's mutation or feature or score. The author provided annotations for the location on the monomeric crystal structure, such as signal peptide for secretation, or buried, or lipid facing, or solvent facing. Click Submit Annotations to look at those features on the structure. By clicking each tab, you can see all mutational hits on the structures with red spears like this, and also average fitness score with colors, and also different notations of the location on the crystal structures. In the study, authors found many solvent-facing hits at the surface of the protein, colored as purple like this, which might be responsible for the protein-protein interactions. MLA-C bind to MLA-D or MLA-A during the lipid transport mechanism, but the actual protein-protein interactions have not been experimentally identified. So in this case, you can get an insight from in silico protein complex structure prediction. So in the paper, authors used AlphaFold multimer to predict MLA-A and C dimeric structures. Instead, in this tutorial, we'll showcase the latest structure predictions such as AlphaFold 3, which you can easily obtain from their own server. Let's unload the predicted dimeric structure from AlphaFold 3 to the interactive mapping module. For the notation, we're gonna map only the solvent-facing hits this time. So let's filter the data and copy and paste again. Now you can visualize the solvent-facing hit, shown as red spears on MLAC structure, highlighted with colors. You can notice that most solvent-facing hits are located around the protein-protein interface, One is at the MLAC contacting with the alpha helix of MLAA, and the other is the C-terminal tails of MLAA wrapping around this MLAC cleft. So in the paper, the author reported that among 25 solvent-facing mutational hits of MLAC, 21 of them lie at the predicted interface with MLAA, highlighting the importance of these mutational hits on the protein-protein interactions. You can apply these steps to any protein structure or any mutational data you have. Finally, please check out the documentation page on the portal to access all of our tutorials. And if you want to leave any comments or questions, feel free to visit the feedback tab. Thank you for watching the video and stay tuned for more.